Now it gets interesting. This is where the genetics that you've already mastered becomes your foundation, your jumping off point. And we begin to talk about more complicated, abstract concepts that are by no means over your head. These are all things that you will easily master, but that still kind of add an extra level of complexity to things. So we're going to jump right in here on the first half of Chapter 7, pages 161 to 176 from the text, and we're going to talk about linked genes today, how recombination plays a role in that, and how we map genes, actually create physical maps of human chromosomes due to recombination and linkage. So we'll start off by defining linked genes. What are linked genes, and how do they differ from everything we've been talking about so far? Talk about recombination frequencies, how we measure recombination, how we spot recombination genetically, and what those numbers mean for us. Then we'll take it all together and say, okay, now that we understand what linked genes are and how they behave, we understand how recombination affects all that, can we go back and do what we were doing with Mendelian genetics? Can we predict the genetics and the inheritance patterns of traits that are linked? And we'll end the lecture with 2-point crosses as a way to map genes, and what that means and what it entails will obviously become clear by the end of the lecture. So, in general, this is what we've been talking about. Let's zoom out for a second and not lose sight of what everything we've been talking about means. So, we're all familiar with double-stranded DNA in general, and probably the adenines, thymines, cytosines, and guanines, although we haven't discussed them yet in this class. I'm relatively certain you've had those in other classes. So we have large, continuous strands or molecules of DNA that wrap up really tightly, like all that rope in my office, if you remember, into chromosomes. Genes are just segments of those DNA molecules, parts of those DNA molecules that are physically in no way different from any other DNA. You can't look at a DNA molecule and say there's a gene and there's not a gene, at least not from its physical structure. You can do that with its sequence, but not with its physical structure. But segments or subsets of these long DNA chromosomes are made up of genes. So these long DNA chromosome molecules have many thousands of genes on them. They're all connected because they're all on the same DNA molecule, and then that DNA is wrapped very tightly around nucleosomes into chromatin to become chromosomes. And you get 2 chromosomes for each chromosome pair, one from mom and one from dad. These figures are showing you the chromosomes after they've already replicated, right? We have 2 sister chromatids here, so don't worry so much about that. When I say you have 2 chromosomes, one from mom, I mean 1 chromosome here and the other chromosome here. And then we call those individual gene segments alleles. You can have a dominant allele from dad on one and a recessive allele from mom on the other. So this is your homologous pair of chromosomes. These are your homologous genes on those chromosomes. You have 2 of each chromosome pair. They can be the same, and then you would be homozygotic for that gene, and they can be different at that gene, different alleles, and you would be heterozygotic. That's what we've been talking about in a very large perspective. And so far, that's what we've been talking about. We've been talking about independently sorting chromosomes during mitosis, meiosis. So each chromosome pair member goes to its own individual gamete with no rhyme or reason to it. It's all random. That's independent assortment, segregation. And the genes we've been discussing, even when we've done dihybrid crosses, have been genes on different chromosomes, physically different chromosomes. So they segregate during meiosis. So this is really what we've been talking about all this time in terms of Mendelian genetics. What I'm showing here are 2 different chromosome pairs. We can say this is chromosome number 1 and chromosome number 14. You have 1 of those chromosome pairs from your mother and 1 from your father in each case. We are looking at 2 different genes, 1 on each of these chromosomes, and in these cases, you are heterozygotic for both genes. You have this allele from your father and this allele from your mother. They're different, so you're heterozygotic. And then on the second gene, you've got this allele from your father and this allele from your mother, and so you're heterozygotic there as well. What we've been saying all along is that you go through interphase, and these chromosomes replicate. So now you have sister chromatids connected. That's all that's being shown here. But now the sister chromatids are identical. They're identical. That's what the DNA replication does. Okay. Then these sister chromatids separate. I ran out of room here on the slide, but they separate, so you can imagine everything doubled. And they're going to go into gametes independently. And so they'll go into gametes in every which way they can with consideration to these two genes, these two alleles. In other words, what I mean is that you can have the yellow allele on one chromosome paired up with the light blue allele on the other. You can have the yellow allele on one chromosome paired up with the purple allele on the other. That's independent assortment. Independent assortment boils down to the fact that just because I got dad's chromosome number 1 doesn't mean I have to get dad's chromosome number 14 too. They can go together, but I can also get a gamete that has dad's chromosome number 1 with his yellow allele and mom's chromosome number 14 with her purple allele. And you get the other combinations too. So every possible combination of alleles that can be represented is represented. That's independent assortment. Every single combination of alleles is equally possible. But now I'd like you to consider this. Same idea, same genes, same alleles, but now they are present on the same chromosome. So we're dealing with a single chromosome pair. You got one from dad and one from mom. You're heterozygotic at both genes, just as before, yellow and red, blue and purple. But now, those two genes, they are physically linked because they are on the same DNA molecule. They are on the same chromosome. So, you do replication, you get identical copies of both, fine. They separate during anaphase. Again, I'm not showing both. You just assume that they're both, both there. I'm not showing the individual sister chromatids that just separated, but that's what these represent. And now we're going to do assortment into gametes. Well, this one's going to go into one gamete, and this one's going to go into the other gamete, and that's it, man. That's all. Where are the other possible combinations? Where is light blue with red? Where is purple with yellow? You don't get those. And the reason you don't get those is because these two chromosomes are physically linked. No other possible combinations are possible because there is no independent assortment. These are linked genes found on the same chromosome. Now let me throw another monkey wrench at you. Same thing, linked genes, same pattern. Here we do our replication here, our interphase. But what happens during meiosis? What happens during prophase 1? What happens when homologous chromosomes are in synapsis next to one another? Crossing over does. Recombination happens, and this is what recombination looks like. It's the swapping of chromosome parts across synapsis. Now when these sister chromatids separate, we get back the original chromosomes we had before, but we also get back the recombinant chromosomes that are the result of crossing over. That's what's shown on the right. And so we can get gametes now that contain all of those combinations. But still, the alleles are linked on the same chromosome. The question then becomes, though, recombination and crossing over is very infrequent. So in the very first scenario I showed you, when the genes were unlinked and we had independent assortment, we got equal numbers of all gametes. In the second scenario I showed you, where the genes were fully linked and there was no recombination, we only got the combinations of alleles that were physically linked on the same chromosome. Now I totally screw things up and I show you that we can get every possible combination because of crossing over during meiosis, but we get fewer of those chromosomes, fewer of those combinations that were the result of recombination, fewer of those that were the result of recombination, fewer of those that were the result of recombination than those that were not the result of recombination, but the original parental chromosomes we started with. So if we get all combinations, but we don't get them in equal numbers, how do we predict genetics? How do we predict inheritance? How do we know how often this recombination happens? How do we measure or predict the frequency of the recombination? And that's really what we're going to talk about for the entire lecture today. So linked genes don't assort independently. Well, duh, I just told you that. They can't assort independently because they're physically chained to one another on a single chromosome molecule. When genes are on different chromosomes, when they are unlinked, they travel independently. They're completely oblivious to one another. We get every possible combination in equal proportions, just as we saw in that first animation. This is Mendel's concept of segregation leading to independent assortment. It results in all the different combinations being possible. If we're talking about two genes, heterozygotic at both genes, that's 4 different combinations, as shown in this diagram from your textbook. So we have an individual who's heterozygotic at gene A, heterozygotic at gene B. The genes are unlinked. We allow meiosis to occur, gametogenesis occurs, and we get all possible combinations. We get gametes that are dominant for A and dominant for B, recessive for A, recessive for B, dominant for A, recessive for B, recessive for A, and dominant for B. That's what I showed you before. It's the same exact thing I was trying to show in that initial animation. And then if we carry this forward and we say, well, what's all the possible combinations of these gametes if these two heterozygotes were to mate, we see that 9:3 to 3:1 dihybrid cross ratio. That's where that comes from. I told you never to use a Punnett square for that. I showed you how not to use a Punnett square for that. But this is what we mean. This is what happens for independent assortment because all 4 possible gamete types are being made. All 4. But genes that are linked, and genes that are specifically linked so that they're close together on the same chromosome, belong to the same linkage group. And so they do not assort independently. These linked genes, they travel together during meiosis because they're on a single chromosome. They arrive together at the same gamete because they're on a single chromosome. They are a package deal, a twofer. Right? You get 2 genes for the price of 1 chromosome. They don't assort independently. See, when Mendel died, some scientists discovered his work, and they realized that he was onto something very, very big. He was onto something that was describing one of the most perplexing mysteries of life, how parents can contribute their own traits in a blended way to their offspring. So other scientists continued Mendel's work using Mendel's model organism, pea plants, to study genetics. And 2 characteristics that were studied by a scientist who came after Mendel were flower color in the pea plant and pollen grain shape. And so the experiment that was done was very much like Mendel's experiment. The scientist took and crossed a pure breeding, so homozygous across the board, a pure breeding, purple flowered, long pollen grained plant with a pure breeding, red flowered, round pollen grained plant. No big deal. We've seen this before. And that scientist got an F1 that had all purple flowers with all long pollen grains. Now, you know and I know right now this is an easy interpretation. All the F1 organisms are heterozygotic, and purple is dominant to red, and long is dominant to round. Then, of course, the individual did the self-cross, the F1 cross to get F2. And what would we expect when we do that dihybrid cross between heterozygotes for 2 genes? We would expect 9/16 of the progeny to be purple and long, 3/16 to be purple and round, 3/16 to be red and long, and 1/16 to be red and round. That is our 9:3 to 3:1 ratio. Instead, the scientists doing this experiment saw this ratio. 284 dominant, 21 dominant recessive, 21 recessive dominant, 55 recessive. What? It flies in the face of everything that was known up until this point. So what happened? Well, this happened. Long and round, F1, heterozygotic, right? Showing the long pollen grain phenotype. And purple and red up top represents the colors of the flowers themselves. Heterozygotic, F1 generation, purple dominant over red. We had interphase, chromosomes replicated. Prophase 1, synapsis, crossing over. Look what just happened. We have now, on a non-recombinant chromatid that did not engage in crossing over, purple flowers with long. On a non-recombinant chromatid that did not engage in crossing over, we still have red with round. But now we have these recombinant sister chromatids that engage in crossing over, where we have purple flowers linked to round pollen and red flowers linked to long pollen. Carry it through. They separate in anaphase, we do gametogenesis, and these are the different gametes we form. So the purple paired with round and the red linked with long are the results of crossing over. As such, they are the rarest of occurrences, because crossing over is a rare event. So they give rise to the lowest number of progeny. The other phenotypes that we see are the highest number are the dominant dominants. It's always the highest number because they are masking the other alleles. And then the lowest number of non-crossing over is the recessive and recessive, as we would expect recessive and recessive to be the lowest, typically. But because crossing over is rare, these mixed phenotypes, where it's dominant with recessive and recessive with dominant, these mixed phenotypes are due to crossing over. Crossing over is extremely rare, and so they give the smallest number of progeny. But we're getting ahead of ourselves. This is too much, too quick. You need to be comfortable with the concept of linkage and how it ties into meiosis and crossing over before we can move forward. And hopefully you are comfortable with that. Hopefully you at least at this point understand what linked genes are and what's going on during prophase 1 and anaphase, during meiosis, what crossing over is and all of that stuff to make gametes. If you're not clear on that, we should meet in office hours or some other time in my office. But if you are comfortable with that, let's go through things a little bit slower and a little bit more meticulously so that you can lock in everything that's going on. Suffice it to say, during meiosis 1, we have crossing over in prophase. And during meiosis 2, we have our separation of sister chromatids and our independent assortment. Those are the two crucial components of meiosis. Before we get into the genetic headache that linkage causes for genetics, let's get some of those basics down. First of all, the notation of linked genes. This would be the most intuitive way to represent a homozygous A, homozygous B individual where the genes for A and B were linked. Here the bar, the horizontal line, represents the chromosome, and they're on the same chromosome, so they're linked on the same chromosome. A cross with a homozygotic recessive parent would be this, showing that the other parent also has A and B linked on the same chromosome but is carrying nothing but recessive alleles. And, of course, the F1 progeny from this cross would be represented like this. Sorry about the little tegrity thing in the way there. This is a lowercase a hiding behind. But this is showing that we have a heterozygotic offspring. Hey, we knew that. Come on now. Let's not insult our own intelligence. But more importantly, the dominant A allele is still physically linked to the dominant B allele because they came on the same chromosome and they stayed on the same chromosome. And the recessive A allele is still paired with the recessive B allele because they came from the same chromosome and they stayed on the same chromosome. So not only is this individual heterozygotic at both genes, we know the linkage pattern. It's dominant A allele with dominant B, recessive A allele with recessive B. A little bit more information coming our way now. So we could represent it with the animations I've been showing you so far that would look like this. So you would never write this, ever. You would never denote linked genes like this. Why not? What is this saying? If you were to denote linked genes in this way, what would it mean? Well, what you would be saying is one, and I'm sorry if I interrupted you if you were about to answer it, but what you would be saying is that one chromosome has on that one DNA molecule a dominant A allele and a recessive A allele. And on the other chromosome molecule, you have the dominant B allele and the recessive B allele. What? Why would you say that? That's not possible. There's only one allele for each gene on a chromosome. The two alleles come from two different chromosomes from two different parents. So this denotion would mean this is going on in the molecule, and that's just never going to be the case. What happens when you have crossing over? Well, crossing over results in this. crossing over flips the alleles of one gene relative to the alleles of the other. So crossing over is when this side of the chromosome is swapped. We usually represent a crossing over event with an X between the genes. And so where it was big A, big B, little b, it is now big A, little b, big B. That's the crossing over. In the end, though, typically geneticists get lazy, and so we usually denote linked genes with a simple slash. So if you had dominant A linked with dominant B and the individual was homozygotic dominant, it would be AB/AB, such as this. The heterozygotic individual that we started with in the example down here would be big A, big B / little b. And you would know that this individual had the two dominant alleles linked together on the same chromosome, and the same would be true for the recessive alleles on the other chromosome. Yeah, okay. So let's solidify some of these concepts by just comparing two extremes. Let's not go crazy into the details yet. Compare the two extremes. The two extremes are completely linked, no crossing over, no recombination, versus completely unlinked. Genes that are completely linked, they're so close to one another on a single chromosome that there's never crossing over between them. That's exceedingly rare. In fact, it's not even realistic. It never happens. But for linked genes, whether they're completely linked or not, we're going to rely on test crosses more than we ever have before. So let's test for linkage between the M and D genes in tomatoes. Let's do this test. We have two genes, gene M and gene D, and we think they're linked on a single chromosome. We're not sure. We're going to test it. M is a gene that encodes leaf shape, normal leaf, not shape, color. And recessive M, encodes mottled leaves. They look dirty and splotchy. D is for plant height, with dominant D being tall and recessive D being short. And we'll take a heterozygotic tall plant, so that's big D, little d, and it has heterozygotic for normal leaves, big M, little m, and we're going to test cross it. What does that mean? Test cross means we cross it with an individual that is homozygotic recessive across the board. So that plant's going to be mottled, little m, short, little d, little d. This is our cross. And if it's completely linked, this is the way we denote the cross. Dominant M is linked to dominant D, recessive M is linked to recessive D. Let's do complete linkage. Alright, so complete linkage, the package deal. Consider first the gametes that you can make. What gametes can be made from these two plants? Now remember, complete linkage, so you can't get every possible combination. This parent has two chromosomes to donate. This chromosome or this chromosome, and that's it. Well, I want dominant M with recessive D. Too friggin' bad. You can't have it. They're not linked together in that pattern, so get over it. You have dominant M with dominant D, or recessive M with recessive D. That's it. And this parent also has the linkage pattern, but this parent only has one type of chromosome to give. So these are the gametes we can make, and the proportions we can make them in. Given that, what are the only possible outcomes of this cross? You can think of it like a Punnett square, actually, because we're not dealing with independent genes. It almost becomes, well, not almost, it does become this. One parent either gives dominant or recessive. The other parent only has recessive alleles to give, and this is the result of your cross. 50% of the plants will be tall and normal. 50% of the plants will be short and mottled. And that's it. Where's tall and mottled? Where's short and normal leaves? It doesn't exist because the alleles were never linked in any parent in that pattern. These gametes are called non-recombinant gametes. Know that term. Be comfortable with these terms. These gametes are called non-recombinant gametes because they are the result of there being no recombination. Non-recombinant, no recombination. And in line with that, the offspring are called non-recombinant progeny, because they are the result of gametes that did not undergo recombination. The best way to understand this, or memorize it if need be, is that non-recombinant progeny, they look like one parent or the other. Right? One parent was normal with normal leaves and being tall, and so is this child. This child has the same pattern or combination of phenotypes as a parent, non-recombinant. And this child is mottled leaves and short, just like the other parent, non-recombinant. Same pattern of phenotypes, same combination of phenotypes as a parent, non-recombinant. Now let's do the same exact exercise, same exact traits, same exact everything, but now let's consider that the genes are completely unlinked. They're on different chromosomes. So now we have heterozygotic M, heterozygotic D, Testcross, totally homozygotic recessive. It's a typical dihybrid cross, like before exam 1, so hopefully everybody's okay with this. Here we have independent assortment because the genes are on different chromosomes. They are unlinked. So every possible gamete is made. We can have dominant M with dominant D. We have recessive M with recessive D, dominant M with recessive D, recessive M with dominant D, all shown here. Not only do we get them all in equal proportions. We get them all in equal proportions. When we cross this with the recessive in our Testcross, the recessive really doesn't contribute much because it's nothing but recessive alleles. And so, as I said before, this parent's alleles shine through. And so we get 1/4 is dominant-dominant, 1/4 is recessive-recessive, 1/4 is dominant-recessive, 1/4 is recessive-dominant. Typical Testcross ratios for dihybrid cross. Here we use terms like recombinant gametes. The big M little d and little m big D gametes are called recombinant gametes. Did recombination occur? No, it didn't occur because these are unlinked genes. But remember, if the definition of non-recombinant is that it looks exactly like a parent, then the definition of recombinant gametes and recombinant progeny is that they don't look exactly like the parents. So this is a non-recombinant offspring, normal leaves and tall. This is a non-recombinant offspring, mottled leaves and short. But normal leaves and dwarf, mottled leaves and tall, there's no parent that looks like that. And so these are referred to as the recombinant progeny, given rise to by the recombinant gametes, the gametes that had combinations of alleles that weren't present in the parent. Okay, so are we okay on all that, I hope? If not, maybe it's a good time to pause the lecture and kind of go back and review, because we're going to move on from here. We're going to introduce the recombination now and see what happens to linked genes when they are not completely linked and recombination occurs between them. So what happens to all this when you are linked but crossing over is possible? What happens to the genetics when you do this? And you don't do it all the time, and you don't do it half the time. You just do it some of the time. We're going to use the same terms. We're going to use the terms non-recombinant to mean the chromosomes that are going to go into the gametes, that are going to make the offspring, that look just like what you started with in the parent. So non-recombinant chromosomes match what the parent started with, i.e. they are not the result of crossing over. And then we're going to have recombinant chromosomes that do not match what was in the parent, give rise to recombinant gametes, which give rise to recombinant offspring. Same idea. If it looks just like one of the parents, it's non-recombinant. If it's a different combination of phenotypes than the parent, either parent, it is recombinant. The question really becomes, in terms of prediction and in terms of the genetics, how frequent is the crossing over? That'd be nice to know. How often does this happen? From that, you can glean how many recombinant gametes are made. That would be good to know. And then with that number, you can start to predict what offspring would look like. With complete linkage, it's easy. Nothing but non-recombinant gametes are made. With no linkage, it's easy. Equal numbers of all gametes are made. In both of these cases, the prediction is no problem. We understand what's going on. But with normal linkage, when recombination can occur, you make more non-recombinant gametes because not crossing over is more common. You make more non-recombinant gametes. Crossing over is kind of rare-ish, kind of, sort of, a bit. So you make some recombinant gametes, but not as many of them as you make non-recombinant gametes. and it gets predicting offspring. Really, really tricky, unless you know what's going on. So let's do the real deal now. Let's go all the way back to our tomatoes. They've been working for us so far. Let's do our tall and dwarf, normal and mottled. Now we're linked, but now crossing over is possible. So we're going to have our heterozygotic with the 2 dominant alleles on one chromosome and the 2 recessive alleles on the other. We're doing a test cross. With these types of experiments, we're always doing test crosses. I'm going to see what happens. So now we get crossing over. It's rare. Doesn't happen often, but we get it. And we get crossing over between the M and D genes. Remember that X. We get crossing over between the M and D genes. And what happens when we get crossing over? One set of alleles flips with regards to the other. So crossing over is going to give us big M with little d and little m with big D. That's what crossing over is going to give us. Sometimes. Kind of rare. Since it's sometimes and kind of rare, most of our gametes are going to be the result of no recombination. Most of our gametes are going to keep the linkage pattern we find in this parent, giving rise to non-recombinant progeny that mimic the pattern we see in the parents, i.e., normal leaves and tall, or mottled leaves and short. Our non-recombinant progeny look exactly like one of the 2 parents. Our non-recombinant progeny came from non-recombinant gametes, which came from no recombination. The largest number of progeny will look like the parents. Crossing over is rare. So the recombinant gametes are small in number. So the recombinant progeny are small in number. Makes sense. But they exist. So the recombinant chromosomes, where big M was with little d and little m was with big D, those chromosomes made it into gametes. And those gametes fertilized eggs, and those gave rise to recombinant progeny that do not look like the parent. Normal leaves and short, not in either parent. Mottled leaves and tall, not in either parent. More importantly, since recombination is rare, recombinant gametes and recombinant progeny are always going to be the minority offspring as compared to the non-recombinants, where no recombination occurred. This is key because you're not going to get the linkage pattern with your questions. You're not going to know how the alleles were linked. You're not going to know if the parent started with big linked with big or big linked with little. The way you're going to determine that, the way you're going to distinguish between those 2 possibilities is by looking at which progeny has the highest number. Whatever those progeny are, that's what the parent started with before recombination. That's your key. That's how you crack the code. And so let's do it, man. Let's do it. What's the linkage pattern? In the non-recessive heterozygotic parent, if these are the results of your test cross. These are the results of your test cross. Now, keep in mind, the answer is not necessarily intuitively obvious because we have all possible combinations. We have dominant green thorax with dominant purple pupa. We have recessive purple thorax with recessive black pupa. And then we have dominant with recessive and recessive with dominant. The question becomes, what's the linkage pattern in the adult that was test crossed to give these progeny? Well, your highest number is your non-recombinant progeny because crossing over is rare. These progeny are those that resulted when there was no crossing over. So whatever the 2 linkage patterns are in these offspring, that's the linkage pattern in the parent. All of these chromosomes came from the test cross parent. We ignore them because the whole point of the test cross was to let the other parent's alleles shine through. So we don't worry about those. We're interested in the chromosome configuration on top, above, on this side of the question. So here, the linkage pattern in that parent was dominant with dominant, because that is the larger number. This is the result of one non-recombinant chromosome, where the two dominant alleles are together. This is the result of the other non-recombinant chromosome, where the two recessive alleles are together. Non-recombinant, no crossing over. That's what the parent started with. Incidentally, we refer to this, and we will refer to this commonly in class, as a coupled configuration. Alleles are said to be coupled when the two dominants are on the same chromosome, as we see here, or the two recessives are on the same chromosome. Same question, same exact question, now with these results. What's the linkage pattern in that parent if this was the results of the Testcross? Well, remember, ignore the bottom row. It tells you nothing. Ignore it. Pay no attention. Look only at the top. Those progeny that have the highest numbers came from no recombination. And so this progeny has dominant with recessive. Non-recombinant progeny, no recombination. This is what a parent started with. The other chromosome was recessive with dominant. The other larger number. These two sets are the result of recombination. You have recombination between these two genes, and you're going to bring the dominant B up next to the dominant P, giving you this recombinant progeny, and the same is true over here. We call this configuration repulsion. Repulsion is when the individual is heterozygotic for two genes, but one chromosome has one dominant allele for one gene and one recessive allele for the other, and the other chromosome has the recessive allele for the first gene and the dominant for the other. That's repulsion. Coupling is when the two dominant alleles are together on one chromosome and the two recessive alleles are together on the other. And it makes a difference. It makes a difference. We're going to see it makes a difference in some of these problems. So you got it? Is that okay? Your textbook kind of breaks this down in this figure. This is everything we were just talking about. But be comfortable with this. Make sure you're comfortable with this. When we're coupled, the dominants are together on one chromosome and the recessives are together on the other. You do the Testcross. So all we're doing is contributing recessive alleles from this parent, and based on which are the recombinant, the non-recombinant like parent patterns, that tells us what was the case before there was any recombination. And then the recombinant progeny that have a different collection of phenotypes compared to either parent, that shows us what happened after recombination. Now to the prediction side of it all. How do we calculate recombination frequencies, and more importantly, what do they tell us? The actual calculation of recombinant frequencies, like mathematically speaking, is very, very easy. You take the number of the recombinant progeny, add them up, and divide it by the total number of all the progeny, recombinant and non-recombinant. You get a number, you multiply it by 100, and that's your recombination frequency. Easy, easy, easy. So let's do it here. I don't even need to do anything. I don't even need to think for this problem. I know that the smallest numbers are always the recombinant progeny. I just told you that. So the total number of recombinant progeny is going to be 8 plus 7, 15. Divided by the total number of all progeny. And that's where most students make their silly mistakes. It's not the total number of recombinant divided by the total number of non-recombinant. It is not that. It is the total number of recombinant divided by the total number of all progeny, which in this case is 123. You do the division, 15 divided by 123, that's 0.122. I did that with a calculator, multiply that by 100. You get 12.2, and that is your recombination frequency. The recombination frequency for this cross, given this data set, the recombination frequency between gene M and gene D is 12.2%. Well, what does that mean? Who cares? What does that mean? Well, it means that given these two genes, M and D, we can expect crossing over to occur during prophase 1 of meiosis 12.2% of the time. And in other words, 87.8% of the time, we will not get crossing over. We will not get recombination. Okay, so I guess that's trivially interesting in some freakish sort of way. But what can we do with that number as geneticists? Well, this is where the prediction comes into play. With the recombination frequency in hand, we can do all of the type of trustworthy predictions that we had been doing before with unlinked genes. We can predict all the offspring and all their ratios from a mating of linked genes and linked traits, just as we could do before, as long as we know or can calculate the recombination frequency. Here we go. In cucumbers, smooth fruit is recessive to warty fruit. So you remember how I broke this down on Show Me. Big T is warty. I should do this on Show Me, but I'm going to solve it for you in a second. But you know how to break this down. And little t is smooth. So same tact as before. Let's just write down the information as it's important. And our second gene is glossy fruit is recessive to dull fruit. Fine. Geneticists have determined that these two genes exhibit a recombination frequency of 16%. So 16% of the time, we're going to get recombination. Suppose we cross a plant that is homozygous for warty and dull with a plant that's homozygous for smooth and glossy. Okay, so let me forget. Don't want to mess that up. All right. So warty and dull, homozygous. Well, I know these genes are coupled because it's homozygous. There's nothing but dominant alleles. Crossed with a plant that's smooth and shiny, or whatever, glossy. So this was the cross just described. What are we going to get from this cross? It's pretty easy. Each parent only has one type of chromosome to give. So this is what we're going to get in the F1. A warty, dull fruit that is heterozygotic, but more importantly than that, with genes that are coupled. The two dominant alleles on one chromosome, the two recessive alleles on the other. Based on the way this question's been worded, we can figure all that out already, right? And that's what's being shown up here. We know they're coupled because of how we got that F1. And then we're going to take that F1 plant and we're going to do a Testcross. Well, we know what a Testcross is too. That means homozygote across the board. What types and proportions of progeny will result from this Testcross? Man, oh man, do I love it. So if 16% is the recombination frequency, that means 16% of the progeny will be recombinant. So let's start with that. If 16% of the progeny are going to be recombinant, what percentage of the progeny are going to be non-recombinant, or just like the parents? Well, the rest. 100% of the progeny minus the 16% that are recombinant leaves us with 84% that are non-recombinant. Great. What different types of parents do we have? So we know 84% of the gametes are going to be like the parents. We know that 84% of the progeny are going to look like the parents. But we actually have two different kinds of parents. We have parents that are warty and dull and parents that are smooth and glossy. So we have to divvy up that 84% equally between those two kinds. So 42% of those offspring are going to get the top non-recombinant gamete, which carries linked, warty, and dull. 42% are going to get that chromosome. But the other 42% of the time, the offspring got the other non-recombinant chromosome, which carried with it smooth and glossy. So the other 42% are going to be smooth and glossy. But those are non-recombinant because they look like each parent. What were we left with? Well, we were left with our 16%. We knew that. That's what we started with. So 16% of the offspring are recombinant offspring. But they're also split in half. And they're going to result from the flipping of one set of alleles relative to the other. So that means 8%, or half of the 16, are going to have the big T with the little d, and the other 8% are going to have the little t with the big D. Because when we get recombination between these two, we flip one relative to the other. Since this is a Testcross, the only alleles and traits that shine through came from the homozygotic parent. And that's our answer. What types and proportions of progeny will result? Well, 42% of the offspring will be warty and dull. 42% will be smooth and glossy. 8% will be warty and glossy, 8% will be smooth and dull. Easy peasy. If you understand the concepts behind it, it gets to be pretty simple to do. If it's not easy peasy and you are hopelessly lost right now, or even somewhat lost, bring it up in your reflection, bring it up in class, come to me in office hours. You know the drill by now. So this is the same problem, again, from your textbook. So the textbook works this problem out. And this is really what we're doing. I'm not going to go through it nearly as robustly here, but just to kind of bring it all together in terms of images. So we've got our heterozygotic parent showing nothing but dominant traits in a coupled configuration, dominant T with dominant D, recessive T with recessive D. Mating on a Testcross with a homozygotic recessive parent showing all recessive traits, also coupled, of course, because there's nothing but recessive alleles. When we do not get recombination, we keep the parental configuration, dominant with the dominant, recessive with the recessive. And we pass that configuration on to the offspring, dominant with the dominant, recessive with the recessive. When we do get recombination between the two, we're going to flip one allele versus the other. And we're going to bring the little d up top and the big D down on the bottom, and we're going to give rise to repulsion-oriented gametes and give rise to traits that are not so much in repulsion, but that don't match either parent. See, this plant is warty and glossy. There ain't no parent that's warty and glossy up here. This progeny, this offspring, is smooth and dull, and there ain't no parent up here smooth and dull. So these are your recombinant progeny. Intuitively, that makes sense. People usually freak out when the numbers get involved, but the numbers are straightforward. When the question tells you that 16% of the progeny have recombination, or in other words, the recombination frequency is 16%, what the question is telling you is that this happens 16% of the time. So if this happens 16% of the time, this is achieved 8% of the time, and this is achieved 8% of the time, because together they make 16. Which means this is achieved 8% of the time, and this is achieved 8% of the time, because it's Testcross. If this all happens 16% of the time, then the rest of it must be no recombination, which is 84% of the time. And if that's 84% of the time, then this is 42% of the time that we make this one, and this is 42% of the time that we make this one, giving rise to 42% of the progeny showing this collection of traits, and 42% of the progeny showing this collection of traits. And these two trait types match one or the other parent, making them non-recombinant. So, what can we do with this beyond predicting offspring? Well, what we can actually do is begin to physically map the location of genes relative to one another on a single chromosome. We can use these recombination frequencies between different genes to determine the order of the genes on a chromosome and their general or relative location of the genes on a chromosome relative to one another. This isn't so important anymore because now we can sequence everything. But before DNA sequencing was possible, this is how genes were mapped on single chromosomes by geneticists. We do a very, very tricky mathematical conversion here. We used either the terms a map unit to represent physical distance on a chromosome, or centimorgan. Same thing. Centimorgan is a physical distance on a chromosome. And here's the mathematical calculation, so memorize this. 1% recombination is 1 map unit, and 1% recombination is 1 centimorgan. So we're going to practice it now. How many map units is 7% recombination? 7. How many centimorgans is 12% recombination? 12. Okay, good. So we've got that calculation down. There are double crossover events. We call these 3-point crosses. This is where genes flip because we have crossing over, but then there's another crossover event later on that flips them back. So what we're talking about is this. We're going to have a recombination event between A and B. It's going to flip these alleles. These alleles are currently coupled. They're going to flip into repulsion, as we see here. But then if we get a second recombination event, also between A and B, they're going to flip back. And see, genes A and B, respect to one another, are coupled again. But we did get some transfer of chromosome material in between. So we can have these double crossover events where we flip and then flip again and kind of restore the original configuration. I bring those up now simply because we're going to go over those in some great detail in the next lecture. I want to open your eyes to it. Again, if we are in a coupled configuration as we are up here, the first recombination event gets us into repulsion. The second recombination event restores us back into being coupled. This makes larger recombination frequencies underestimated when we do these calculations, but let's worry about that in the next class. Just be aware that this occurs and have some general understanding of what a double recombination event is. And like I said, we'll come back to that in the next lecture. So let's do some mapping. We've got some map units here, same as centimorgans, same as recombination frequencies. Gene A and gene B are 5 map units apart. Gene B and gene C is 10 map units apart. Gene A and gene C is 15 map units apart, and we want to use this information to create a map. It's pretty, honestly, straightforward and intuitive. Let's say, for example, my house is 5 miles from Rinaldi's. Rinaldi's is 10 miles from Bay Path. My house is 15 miles from Bay Path. That's all we're saying here. That's all we're doing. So we would construct a physical chromosome map that looks like this, given the information on the left. This map makes all that information make sense. And so this is the physical map for those 3 genes. Let's do another one together. Gene A and gene D are 8 map units apart. Gene B and gene D are 13 map units apart. Gene C and gene D are 23 map units apart. Pause the lecture for a second. Leave this on the screen. Get out a piece of scrap paper and see if you can construct the map. The map looks like this, I hope. This map unit configuration has everything make sense. Now, you might have done this differently. And if you did, go back and look to see why it might be different. But you're putting these genes together in an order that makes some sense. And from that, you can have predictions. This value comes from the data I provided. This value comes from the data I provided. And this value comes from the data I provided. If this was an experimental case, if this was an experimental exercise, you could do some test crosses where you compare gene A to gene B. Your map predicts quite robustly that you should see 5% recombination between gene A and gene B. You could do test crosses to measure that. Your map also predicts 10% recombination between gene B and gene C. You could test that with recombination frequencies. These are genetic maps, and you may in the future be called upon to construct such a map given recombination frequencies. It's like a jigsaw puzzle. You're really just mixing and matching the lengths until you get a map that makes sense. 2 important and key points to make before wrapping up this portion of the lecture. Unlinked genes assort equally and independently. I won't go through the whole exercise of it, but that is 50% recombination. 50% recombination means that half the time you get one configuration and half the time you get the other. That's unlinked. That's what unlinked genes do. 50% of the time they go in one and 50% of the time they go in the other. They're equally proportioned. So that's one. What that means, though, to us, physically speaking, is that any 2 genes that are on the same chromosome, so yeah, they're physically linked, they're found on the same chromosome molecule, but they're 50 centimorgans apart or more, they behave like unlinked genes. Why? Well, they're so far apart at 50 centimorgans that recombination occurs in between them as often as it doesn't. What that means is that we get the recombinant gametes in equal numbers to the non-recombinant gametes. Well, getting the recombinant gametes equal in number to the non-recombinant gametes, that sounded a lot like the unlinked genes, and in fact it is. So once two genes get further than 50 centimorgans apart, once recombination frequencies hit 50%, we are at independent assortment. Whether they are physically unlinked or just so far apart on the same chromosome, we treat them like unlinked genes because they assort independently. Coupled with that idea, kind of intuitive in all that, crossing over gets more and more likely, more and more frequent as genes get further apart on a chromosome. Well, actually, we already said that without saying it. What is another way of saying 6 centimorgans? 6% recombination. Recombination occurs 6% of the time. What's another way of saying 25 centimorgans? 25% recombination. Recombination occurs 25% of the time. So yes, of course, as chromosome distance goes up in centimorgans, recombination frequency goes up because the two are the same. And so recombination gets more likely. Also, as two genes get further and further apart on a chromosome, double crossovers occur more likely, occur more frequently, restoring that parental non-recombinant linkage pattern, as I showed you already. So things to keep in mind. So now let's consider this set of data and try to come up with a genetic map that explains all this. So I want you to do that. Again, pause the lecture, indulge me, get back to that piece of scrap paper and see what you can come up with here. Well, gene A and gene B are unlinked. We see that. Recombination frequency of 50%. Recombination frequencies never go above 50% because 50% is equal 50-50. So 50% is the highest recombination frequency we ever see. Gene A and gene B are unlinked. Are they on different chromosomes? I don't know. Are they just really, really far apart on the same chromosome? I don't know, and I don't care. It doesn't matter to me, the geneticist. They are unlinked. They assort independently. Gene A and gene C are unlinked. Gene A and gene D are unlinked. So A is the odd man out. A is not linked with the rest. Gene B and gene C are 20 centimorgans apart. Gene B and gene D are 10 centimorgans apart. Gene C and gene D are 28 centimorgans apart. It sounds to me like C and D are on the ends, B is in the middle, and A is on another chromosome. And that's exactly the linkage pattern for these 4 genes. Alright, so what did we talk about in this lecture? Well, we said when genes are on different chromosomes, they travel independently during meiosis. And we've been saying that since the second lecture, segregation and independent assortment. We've seen the results of that in our dihybrid cross lecture, where we get the 9:3 to 3:1 phenotypic ratio when we cross 2 heterozygotic individuals in a dihybrid cross. Genes that are located close together on the same chromosome are referred to as linked genes. They are physically linked on a single chromosome, and they travel as a package deal during meiosis. When genes are completely linked, they're on the same chromosome and we never have recombination between them. All we get are non-recombinant gametes, giving rise to non-recombinant progeny. These are progeny that look like one or the other parent. But that never happens. I'll tell you right now, that never, ever happens. You always get some recombination. So when you get recombination, you get recombinant gametes. These are allele combinations that were not present in either parent. These gametes give rise to recombinant progeny that are also unlike either parent. But these recombination events are rare. They don't occur frequently. Therefore, recombinant gametes are rare, recombinant progeny are rare, and so they are always the minority in the progeny. Recombinant progeny are always fewer in number as compared to the non-recombinants that came by no crossing over. This is how you spot which progeny are the result of no recombination and which progeny are the result of recombination. More importantly, this is how you will always be able to determine which configuration of alleles is recombinant and which is not. And the terms you will use to discuss and describe those configurations are coupled, when the dominant alleles are together, and repulsion, when the dominant allele of one gene is together with a recessive allele of another. We discussed the calculation of recombination frequency. You take the number of recombinant progeny and divide it by the total number of all progeny, take that decimal, and multiply it by 100. And then we worked through some really easy problems. We kind of talked this through. We were given a recombination frequency of 16%. We realized that that meant that 8% of the offspring had to be from one recombinant chromosome, the other 8% from the other recombinant chromosome. It left us with 84% of the progeny being non-recombinant. That meant 42% came from one parental chromosome, the other 42% came from the other parental chromosome. We talked a little bit about mapping. We made the point that map units and centimorgans are equal in number to recombination frequency, and we talked about how you can take these map units to construct and decipher a physical map of a chromosome. I almost said human chromosome, but it's true of any chromosome. Now in the next lecture, we're going to continue to build on these concepts, and we're going to talk about what happens with linkage, recombination, and mapping when you are so far apart that you do get a lot of these double crossover events. They're called 3-point crosses. So the next lecture is going to be all about 3-point crosses. You should probably not move on to the next lecture if a lot of this material was confusing to you. If this material was very confusing to you, you have many options in front of you. Reread the notes, use the textbook, look at some of the sample problems in the textbook that are worked through, review the portions of this lecture you found confusing, but most importantly, seek my help in office hours or tutoring help at the tutoring center. I do not recommend you build your knowledge on a house of cards, meaning if you're not clear on this, don't try to move up into the even more difficult aspects of this. Make sure you fortify the knowledge that you need here before you move on and build on it in the next lecture. But as always, thanks for watching, and we'll move on to 3-point crosses next.